Hello, this is Chen from Baylor College of Medicine. Today, I'd like to take the opportunity to give you a quick update on the single-cell multi-omics atlas of the retina. So one of the most exciting technology breakthroughs in the last decade in biomedical field is the single-cell omics technology. It has made a tremendous impact on the biomedical research. So since the initial publication of single-cell RNA-seq technology back in 2009, the technology have gone through many iterations of improvement in both its chemistry, the instrument can be used, and also the protocol for performing such profiling. As a result, we see a dramatic reduction of the cost per cell that reduced from initial about $7 per cell to currently about 1 cent per cell. The throughput has been dramatically improved as well, and as a community together, we are approaching about 100 million cells has been profiled by the community. Concurrently, the number of publications use this technology has been seen dramatically increase to thousands of papers in the last decade. So one of the most commonly used technology is a commercialized technology is coming from 10x Genomics, as people are familiar with. It's built on this droplet single-cell technology that is initially published in the landmark paper in Cell in 2015. As cartoon here, the job sequencing is utilize the microfluidic system where this one of the channel contain the cell lysis buffer and the small beads flow through one channel in the aqueous channel, while the cell are loaded in oil for a different channel. When these two phases meet, they form a small droplet, and that can be put into a tube. So based on the Poisson distribution, double Poisson distribution, most of the droplet will be empty or have a maybe beads only or cell only, with only a small fraction of the droplet have one bead and one cell. So in such droplet, the cell will be lysed by the lysis buffer, and the RNA released will be captured and bind to the oligos on the surface of the bead. So the oligo is specially designed have a oligo DT that's captured the poly A tail of the mRNA, but also has a unique barcode for each bead. That's what we call barcode, the bead barcode, and then the very end of the oligo is a universal primer that can be used to amplify the cDNA later on. So once the droplets are break into a homogeneous solution, and then the reverse transcription can happen, and with the using the RNA that capture on the beads as a template and form the first strand cDNAs molecules. And there's a reverse template switch method that will allow you to add a universal primer at the end of this first strand cDNA. So after the first strand cDNA synthesis, and then the PCR will be performed to form the full-length cDNA, and that can be later on sequenced with next generation sequencing method. Molecules that carry the same beads barcode RNA come from molecules from the same cell, and that can be grouped together after the sequencing, and to basically identify or profile the transcriptome profiling of that cell. So with this method, it's a super high throughput because we can generate thousands of such beads in a matter of 5 to minutes, and then the whole reaction is actually done in the one tube. So it's super high throughput. So with this type of technology, it's now then possible to not unlike before where we do bulk profiling, where the cells from the same tissue are homogenized together, and for analog this to this picture that they form a sort of smoothie. So what you measure is the average of all cells in that tissue. And now with this technology, you can identify and profile individual fruit go into that smoothie. So with the single cell technology, it's really it's really ideal for dealing with these three challenging issues or answer this three type of question. One is you need dealing with when cell heterogeneity is a challenge, or so instead of to measure the average measurement on the bulk samples, it allows you to discover the true diversity within the sample, such as different cell types and clonal tumors and that nature. Also, if the research is interested in particular rare cell type population in the tissue, that is they don't have proper marker to identify or purify, and then this single cell technology allow you to measure the profiling omics profile of those rare cell type population easily. And then lastly, when you combine with the functional assay, the single cell approach can allow you to perform very high throughput single cell perturbation type experiment to measure the perturbation at the individual cell level. With the progress of the single cell omics technology, the international committee launched the project called Human Cell Atlas. With this project, the goal is to create comprehensive reference map of all human cells as the base for further understanding of human health and diagnosing, monitor, and treat disease. So our group and other groups work together and as part of the Human Cell Atlas project, with the goal to build the single cell atlas reference map for the eye. So as we know, the retina of the human eye is very highly cell heterogeneous. From the last several decades of study based on the morphology and function, and we know there's many cell types in the retina, and it has been estimated there's in human retina there are 4 photoreceptor cell types, 2 horizontal cell types, 13 bipolar cell types, about 30 amacrine cells, and 15 ganglion cells. Together about 60 some cell type based on morphology and function. So in the current single cell atlas reference of human retina based on single cell eye technology, there's a total of about more than 2 million single nuclei or single cell from the retina has been profiled. So there are 2 maps. One is composed of the single eye sequencing data containing about 1.8 million nuclei. The other is about 250,000 single cell RNA-seq. So in terms of the cell type, these 2 methods produce very similar results. And then, however, when you look at particular gene expression, these 2 methods have a different bias. So depends on the data you are the data you generated, you may use one or the other or both reference. So totally currently there's more than 2 million total number of single cell nuclei from 50 different donors, and over 110 cell types have been identified. So currently there are 14 bipolar cell clusters identified in this current map, which is one more type before compared to the previous map. And because with the increased number of cells in this current map, the blue bipolar and giant bipolar can be clearly separated out into 2 clusters, and previously it's like 1. So the big increase of the cell types identified in the current cluster, the map come from the amacrine cells. So instead of 34 clusters in the previous study, currently there's 674 amacrine cell cluster identified. For the RGCs, it has been increased from 9 clusters to 15 clusters, which is the similar number identified from previous morphology study. So with the single cell atlas reference in hand, there's many utilities, including, for example, as a list, the cell type identification. As I mentioned, there's more than 110 cell types identified in the retina. We can identify gene markers for each cell type. We can check the expression of each gene across each cell type. And it also allow you to perform differential gene expression analysis to identify gene and gene sets that show specific expression in different cell type. And then we can also contrast co-expression gene network across each cell types. So to access this data set, we can go through the resource page from our lab. You can also just scan the 2D barcode and to get to the website. So once you have this data portal website, you can click on the Human Cell Atlas link, and it will bring to multiple links that this data set has been deposited to, that allow you to download and browse and also perform some interactive actions. For example, one of the places we have deposited and organized the data is at the Cell by Gene and portal at Stanford. And here's the landing page you will see for the Human Retina Atlas. It's a different color, represent different cell type. And then this how to use this tool and explore this data. You can find a more detailed tutorial in this YouTube link. And then also can find that link when you go to the data portal. Similarly, the exact same data has been also deposited to the UCSC genome browser, that you can look at not only the gene expression, but also the ATAC-seq data that come along with this data set. So here's the link to the UCSC browser, and also here's a YouTube tutorial for how to use this data, explore this data on UCSC browser. So in addition to the Retina Atlas, we have also in the process building the atlas for the rest of the eye part, including ciliary body, trabecular meshwork, limbus, sclera, lens, iris, cornea, and then also the RPE. So in addition to the gene expression, we're also quite interested in how genes are regulated. So one important information one needs to gather when it comes to gene regulation is looking at its as chromatin structure. So one of the approach to looking for the open chromatin structure in a cell is through the transposer accessible chromatin assay, ATAC-seq for short. So the principle of ATAC-seq is as follows. So the chromosomes are packed in the nuclei through the nucleosome organization. Some areas are densely packed, where the open region are often the gene-regular region. So it has been observed these open chromatin regions are more accessible by the hyperactive transposase homodimer. So when you incubate the chromatin with the transposase, it will preferably inserted in the open chromatin region, as this adapter bring and inserted into this location. So the transposer element insertion can then be used to amplify the fragment that's attacked by those transposes. And after sequencing, when you map back the reads back to the genome, they will highlight the regions that is more accessible or more open in the data. So here's a quick summary of the single nuclear ATAC-seq generated in the map. So from 22 donors, 150,000 nuclei have been profiled, and you can see in the UMAP, based on the chromatin accessibility profile, it can all the major cell type in the retina can be readily identified. So compared to the single cell ataxic data to bulk ataxic data had been generated before, as we can see, vast majority of the regions that identified from bulk ataxic are included in the single cell ataxic. But moreover, the about tenfold increase of the chromatin accessible region have been identified only by the single cell or single nuclear ataxic data. The reason of this increased sensitivity is because twofold. First, single nuclear ataxic seems more clean. It seems cleaner or more sensitive and have a higher signal to noise ratio compared to the bulk. This is one example. PD6A is a gene that highly expressed in the rats. As you can see, the promoter region. This is the bulk data. You see the promoter region depicted by the red arrow, and the enhancer region depicted by the green arrow. So you can clearly see a promoter peak in the bulk data. However, the enhancer region, the signals were low. But when you compare to the single cell data, and then you can see in the raw cell type, a very strong promoter region, and then you have a lot of signal in the enhancer region. In contrast, other cell types where the gene is not expressed, there's very little promoter information signal, but also almost none signal in the enhancer region. So this increased or improved signal to noise is more obvious, even more obvious in genes that are highly expressed in the rare cell type. For example, SLC6A1 is a marker gene for mural glial cells. As you can see, the promoter region of mural glial cells is a very high signal, and while in the bulk, essentially no signals, only noise there. And then you can also see the enhancer signal in the mural glial cells are very high, and it's a little bit in the apical cells, but pretty much absent in other cell type. So the single nuclear ataxic data can be integrated with the single nuclear RNA-seq data to generate this multi-omics cell atlas. So as you can see, this is the co-embedding between ataxic and RNA-seq data. And you can see they align really nicely. When we examine the marker genes of a different cell type, you can see they can all have very clean and very strong signal across the diagonal, very specific. The embedding of integration between nuclear RNA-seq and ataxic can not only happen at the the class level with lower resolution, but it can be done in a much higher resolution. This is an example for bipolar cells, and you can see the co-embedding of them. They go very nicely together. So with the multi-omic data set, then you can now start link the individual peak or open chromatin region to its potential target gene through either the co-accessibility between individual peaks with the promoter region of the gene, or through the peak-to-gene correlation, where the openness of the peak and correlate with gene expression to identify peaks are correlated strongly with the gene expression. So through this sort of linking, the current atlas version have about 670K open chromatin region identified, and this is the distribution of those open chromatin regions across different cell type. And then among them, about 20% show cell type specific or differentially accessible region, and then suggesting they might be responsible for cell type specific gene regulation. So with the open chromatin profile and the RNA-seq integrate together, and then we also can add another modality based on the genomic sequence, genomics of the individual donor. On top of this, this will open the opportunity to do a series of sophisticated analysis. In addition to the gene peak association, as I mentioned before, you can also now looking at single cell allele specific chromatin accessibility, single cell level, different cell level of computing accessibility QTL, and single cell level of allele specific expression, single cell eQTL study to link the individual variant to the gene expression and then the open computing accessibility. But in addition, by overlay with the GWAS results, you can also perform gene set cell type enrichment assay and also perform fine mapping of GWAS locus. Just to give you an example, take the gene expression profile obtained from the single cell data, and then we overlay with the GWAS trait, published before, and then you can see the y-axis are different trait, and then the x-axis is the different cell type the different cell type, and you can see the enrichment of AMD cell type, for example, only observed in the adult retina and RPE tissue, while there's many more cell types that show improved enrichment, gene set enrichment in the glaucoma trait, and also when you look at the trait for the retina, and you can see the major cell type that get enriched are cone cell and rod cells, which makes sense. So through this exercise, you can identify or prioritize cell types that linked to particular trait. Similarly, this similar assay has been done for other parts of the eye, and you can see there's hardly any cell types show enrichment for the AMD related gene set, and you can see there's many other cell type identified for the glaucoma related trait, for example, like ciliary, and then the trabecular meshwork cells, some cell type in the trabecular meshwork, and again for the retina trait, mostly coming out from the photoreceptor cells and then the sometimes the neuroprogenitor cells. So then you can also further zoom in to look at this enrichment, look at what gene are driving this sort of enrichment. For example, in the RP, which are enriched for the GWAS, for the AMD GWAS gene set, you can see that those other genes are driving the enrichment value. So we can actually go beyond just the eye atlas, since there's many cell atlas have been generated for different part of the human body, and we can perform the cell type enrichment exercise across the entire human body. And then this is some preliminary results we have done, and you can see the top cell types that enriched for the AMD. In fact, almost the eye is not ranked very high. Then you can look at the cell types actually across whole human body that enrich for AMD GWAS hit. You can see actually they are primarily driven by immune cells. So in contrast, for the open angle glaucoma GWAS hit, the eye is indeed the top tissue type that's enriched for PLAG GWAS gene sets. And when you look at closely what cell type are driven this enrichment, a lot of them are epithelial, endothelial related cell types that show up. So with the multi-omic data, you can also perform fine mapping of the GWAS loci. And this is just one example. The TGF-beta receptor 1 is a gene that can be identified as one of the AMD GWAS loci gene. And when we look at the expression in the retina, this gene are only very specifically and highly expressed in the microglia cells. And then when we then look at the the SNPs that have been linked to the have shown association with the AMD patient, and there's many actually SNPs in the LD, and then we overlay that with the open computing profile, and in particular in the microglia cells, and then in fact and perform the posterior inclusion probability test, and this particular SNP actually show up as the strong candidate for the causal SNP. So the similar exercise can be done for all loci, for all cell types, to prioritize potential causal variant and also the cell context this variant plays in across the all cells in the eye. So quick summary, in the current human cell atlas reference, this includes single cell RNA-seq and ATAC-seq, and then with over 110 retinal neural cell types have been identified. So this is significantly higher than previous estimation of 64 cell types. Integration of single cell or single nuclear RNA-seq and ATAC-seq enable one to systematically characterize the gene regular network, and then by overlay this with genomic data, the impact or the effect of genetic variants in gene expression and chromatin accessibility in each cell type can be assayed. So the human cell atlas project is still ongoing. So in terms of the eye, the current ongoing projects or goals is to include into the atlas of donors from diverse ancestry groups. The current data are primarily coming from white donors, and we are also in the process of to extend atlas to include pediatric samples, and also to start to include data set from patients with various diseases such as glaucoma, AMD, and DR, diabetic retinopathy, and also as I showed you, the process we're in the process to complete atlas to include all parts of the eye, and also to increase the number of donors to enable more robust QTL type study. And lastly, we are in the process to extend the atlas to include additional modalities such as spatial transcriptomics, metabolomics, proteomics, etc. So which will take many years to complete. So just to give you a quick example of spatial transcriptomes. So I said back to the analogy of the fruit, in the bulk sequencing, it's like we are measuring the average from the smoothie, and the single cell allow you to identify individual fruit going into this bulk smoothie. And however, during this process, the spatial relationship among those fruit are lost. So given the importance to get the spatial information for many research region, and a large number of protocols or methods have been developed, including both sequence-based and hybridization-based method to identify or to perform the spatially resolved transcriptome profiling at single cell level. So our group have successfully adapted Murphy's method to the retina. As I can show you here, instead of this is the data generated from the Murphy's spatial profiling of the mouse retina. So in addition to this sort of UMAP, you are familiar with, we can identify all the major cell class in the UMAP, but now you also can place individual cell back to the tissue, and as you can see, this beautiful layer structure of the retina. So you can learn more about this data by going to this website to scan this QR code and then learn more. And we are in the process to apply this method to generate a spatial resolved human retina map for the community. So with that, thank you very much for your attention, and I hope to give you more update on the spatial transcriptome front in the future.